Hello, and thank you for joining us today. Welcome to our webinar entitled Streamline cGAS Inhibitor Discovery with the Transcreener cGAS Assay. I'm Justin Brink, Director of Marketing here at Bellbrook Labs. I'd like to introduce to you our presenter, Meera Kumar, Senior Scientist at Bellbrook. Meera has an amazing array of experience, from developing assays like this to our drug discovery programs. She is well versed in strategies for discovering, developing, and advancing small molecule inhibitors for enzyme targets. I'd like to note that Meera and I are working in separate locations due to the coronavirus. I hope you are all staying safe during this unprecedented time. Thank you. And with that, here's Meera.
Hi, good morning or good afternoon, everybody. Thank you, Justin, for introducing me. The agenda for today, we will first talk about the rationale for targeting cGAS, why it's important, especially with respect to the autoimmune disease. Then we will move on to the steps and strategy that we undertook at Bellbrook Labs to develop a Transcreener cGAS assay. And then we will briefly look at what sort of applications that you could run with this assay, and then have a conclusion or summary slide. And then we'll have a Q&A session. And just in case if we are unable to get to all your questions this afternoon, please do send us an email and we will respond to any technical questions that we are unable to cover at this time. So with that, let's get started. So the innate immune system relies on pattern recognition receptors, abbreviated as PRRs, to recognize the pathogen-associated molecular patterns, PAMPs. So that could be like the viral particles, dead cells, etc., etc. So upon recognition of a PAMP, the PRRs generally they generate a signaling cascade that would lead to transcription of genes that's associated with the immune response. Now, because all pathogens actually utilize nucleic acid, either DNA or RNA, the PRRs then recognize those nucleic acid to trigger the immune activation. So in normal cells, our DNA is confined to the nucleus or inside the mitochondria. The presence of DNA in the cytosol is therefore indicative of either cellular damage or infection, and that leads to activation of genes that's associated with the immune response. One way the cytosolic DNA is sensed is through the cGAS STING pathway, specifically by this enzyme called cGAS, cyclic GMP AMP synthase. So when double-stranded, the cGAS gets activated by double-stranded DNA. So when double-stranded DNA binds to cGAS, or cGAS recognizes double-stranded DNA, it dimerizes and stimulates the formation of cGAMP. So in presence of ATP and GTP, it forms a second messenger cGAMP. This cGAMP then directly binds to STING. STING stands for stimulator of interferon genes, which triggers phosphorylation of several partners, primarily the transcription factor IRF3 via TBK1, and that promotes transcription of inflammatory genes like the interferon beta. So that's the typical pathway. cGAS has been implicated in roles in tumor surveillance. So as you can imagine, in tumor cells, there is DNA damage and there is a lot of accumulation of cytoplasmic DNA. So there is a role that cGAS STING pathway plays in activation of IRF3 for tumor surveillance. There is also a role that it plays in autoimmune diseases. So for example, an example is a disease like AGS, where there is chronic activation of STING due to host DNA in the cytoplasm, and there are also mutations in the TREX1 protein. It's an exonucleus. If there is a mutation in there, it causes endogenous accumulation of DNA in the cytoplasm, which can again lead to the activation of the cGAS STING pathway, resulting in interferon production. So therefore, a good strategy would be an inhibitor for cGAS for all these autoimmune diseases. So we decided to develop an assay here at Bellbrook Labs, and based on the Transcreener platform. So probably some of you are already very familiar with the Transcreener platform. For those who are not, I will briefly go over the principle of the assay. So we always start by raising a monoclonal antibody against the nucleotide, in this case cGAMP. And that's the monoclonal antibody, one of the detection reagents that you see here. And the other detection reagent is nothing but cGAMP that is conjugated to a fluorophore. We have two different fluorophores. We have this in two different flavors, actually. One is the fluorescent polarization assay, and the other one is the TR-FRET assay, which I think we are just introducing that. So that's a very new assay. In both cases, we take cGAMP and it's conjugated to ATO dye. It's slightly different for the two tracers. For the FP, we use ATO 633, and for the TR-FRET, we use ATO 647. So how does this assay work? So let's pretend we're starting with enzyme reaction. You add ATP, you add GTP, you add double-stranded DNA, you activate your enzyme, and you produce cGAMP. So at time 0, let's say that no cGAMP is being produced. This antibody is bound to the tracer. Okay, so this tracer is now, since it's bound to a bigger molecule like an antibody, it restricts and hinders its rotational ability. So it cannot tumble very freely. And therefore, you register a very high polarization value. Now, as the enzyme starts churning out cGAMP. Now, cGAMP has a very high affinity for this antibody, so it kicks off the tracer, which is now free, and cGAMP binds to the antibody in its place. Now, this free tracer can just rotate freely. There's nothing hindering its movement, so it changes the polarization. So you end up with a low polarization. So you begin with a high polarization, and the polarization decreases as you produce cGAMP. The TR-FRET is very similar. All that we have done is we have taken the native antibody and we have attached it to terbium, the lanthanide, and we just excite the terbium. And when the terbium gets excited, it transfers its energy to the tracer because it's in close proximity, and that would excite the tracer, and we would measure an emission. So if in the absence of cGAMP at, let's say, time 0, there is a FRET because of proximity. Now, as cGAMP is produced, it kicks off the tracer and the FRET is removed. When you excite it, it's not able to transfer its energy to the tracer molecule because it's farther away. So both these assays are kind of a negative output assay. You begin with either a high FRET or a high polarization, and as the time progresses or as you build the product, you would see a decrease in cGAMP, and in either polarization or the ratio. And this would become more clearer as I show the data. The assay itself follows all our Transcreener assay protocols. We wanted to make it super simple. The example here is low volume 384 well, and we start the process or the enzyme reaction in 10 microliter volume. And you can split that 10 microliter volume depending upon your automation capability, your liquid handling capabilities. You can do a 7.5 microliter enzyme reaction and add 2.5 microliter of substrate. Just keep in mind that the 2.5 would be 4x the concentration because you're adding it into a 10 microliter reaction. You can do it 5 plus 5. Either way, you start with the enzyme reaction, which has cGAS, ATP, GTP, and double-stranded DNA. And then you incubate the plate for about an hour, or, depending on what concentration of your enzyme you choose, you incubate for a certain period of time, and then you quench the reaction by and also add the tracer and the antibody, the two components of the detection mixture, and mix the plate and read the plate. The equilibration time is pretty fast with this assay, about 5 to 10 minutes, which also helps us read this assay in real time if that's And that's one of your goals. And we will show some of the applications where we need to run it in a kinetic mode or real-time mode to get some off rates and whatnot. But basically, this follows the simplistic homogeneous. It's a direct assay. It's a homogeneous assay, but it also follows the simple HTS ready mix and read format. Okay. Now, even though at Bellbrook we run all our assays in low volume plate in 384 well format, the assay is compatible with 96 or even 1536 format. If you're running it in 96 well plates, we suggest that you use a half area plate, which then you split your reaction as 25 microliter enzyme reaction and 25 microliter detection reagents in a total volume of 50 microliter. That would be for 96. If you choose to run it in 1536, you can choose split it as 3 microliters and 3 microliters with a total of 6. I mean, you could do variations of that. It's a pretty flexible assay. We give the user the flexibility to design the assay the way you choose to. So now we'll talk a little bit about assay development, what went into developing a cGAS assay. And the first thing was to see how selective the antibody was. And as you know, for a cGAS assay, the two nucleotides that we primarily use are ATP and GTP. So we wanted to first rule out if this antibody would bind to ATP or GTP. And as you can see, it has no cross-reactivity towards ATP or GTP. We also put in other nucleotides, like closely related nucleotides like ADP and GDP, D-ribose, cyclic AMP or cyclic GMP, and none of these nucleotides cross-reacted with the cyclic GAMP antibody. That was great. Then we took the 2,3-cGAMP, which is the product of the cGAS reaction, and you see you get a beautiful sigmoidal curve. But we also noticed that the antibody can identify the 2,3-di-GMP with the exact, almost same affinity as 2,3-GAMP. So we did see that, but the linkage is very important because when we tested 3,3 cyclic GAMP, you can see that the antibody does not recognize the linkage 3,3, but specifically recognizes the 2,3 linkage. So that was good to know. With that, we decided, okay, let's run a cyclic GAMP standard curve and see how it pans out. Now, once again, people who are familiar with Transcreener assays in general, say if you have used a Transcreener ATP assay or AMP assay, we typically give you a starting antibody concentration. If your initial ATP is, say, 10 micromolar or 100 micromolar, you might need to adjust your antibody concentration. And we need to do that because those antibodies have some cross-reactivity with ATP and GTP, and that's why we need to adjust the antibody concentration. But as I had shown in one of the previous slides, the cGAMP antibody does not bind to ATP or GTP, and therefore running a standard curve is really easy. All you do is just titrate the cGAMP itself in either FP assay or in TR-FRET assay, and then this is the polarization value. So you can see as we increase the cGAMP concentration, so for 100 micromolar, your polarization decreases compared to at no cGAMP. It's a higher number, and it's the same for TR-FRET. It decreases as you increase the cGAMP, as I had mentioned. And you keep the ATP and GTP constant, just so you know you're mimicking the enzyme reaction. So let's take a look at the Z prime, and this is reading the plate after an hour. The lower limit detection is about 0.2 micromolar in both the assay formats. Lower limit of detection is defined as the first appearance of a positive Z prime, at about 5 micromolar, you get a really good Z prime, and that's pretty much what we would expect when you would run the assays starting under the standard conditions. So you can see in both the assays, you get a really good Z prime at about 5 micromolar. Now, plate stability. Plate stability is reading the plate after 24 hours. So if you're running, say, a million compound screen and you don't have time to read the plates right away, you can come back the very next day and read the plate and still get a decent Z prime at 5 micromolar. I would like to, the TR Fred does take a slight hit on the Z prime. You can see even at the highest from 0.95 to 0.77. The lower limit of detection almost maintains. It goes down to about 0.4, but that's the difference between, the native antibody versus an antibody that's labeled with terbium. So just a little bit of caution there, but still at around 5 micromolar, you get really good, decent Z prime. So let's move now to enzymology. So we produce cGAS. We also sell human cGAS here and at Bellbrook. So this is a titration of cGAS. So a complete reaction is where the cGAS gets ATP, GTP, and double-stranded DNA. And the concentrations that we used are 100 micromolar ATP and 100 micromolar GTP and 60 nanomolar double-stranded DNA, which is a 45 base pair ISD, the interferon stimulating DNA. And in absence of substrate, you see a clean line. You don't see any this just points to the purity of the enzyme, that there is no contaminant that's actually displacing the tracer from the antibody. Now, the point to note here is our assays, or just not our assays, any competitive binding assays are by nature nonlinear. What it means is you will get a sigmoidal graph when you plot any competitive binding assay. It's not just Transcreener assays. It's not just assays that Bellbrook Lab makes. It's any competitive binding assay. So, so how do you convert that into a linear scale? So you could take the raw data, which is polarization values, run a standard curve that I just showed in the previous slide with ATP, GTP, and double-stranded DNA, and convert those polarization values into the product formed, which is cGAMP, as you see here. And you get a really good R-square value. And let's just look at anywhere between 0 and 10 micromolar, which is from, this part of the curve that you see. And it looks really non-linear, but if you convert it with the standard curve, you can see that it's very much linear. And that's how, if you don't need to do it, but if you have to get further enzymology, like if you want to determine Vmax, Km, etc., you will need a standard curve. But you don't need to do that for regular dose response curves if you don't have to. And we calculated the rate, and here we are defining the rate as nanomolar cGAMP produced per nanomolar cGAS per minute. And I know typically it's given in terms of nanomoles, but we've just defined it that way here. And it's close to 1 nanomolar cGAMP produced per nanomolar cGAS. Now, Z prime determination, we're showing a couple concentrations here, 5 nanomolar, 10 nanomolar, and here is, in the absence, it's again cGAS, but no ATP or GTP. And this gives a good window for you to unambiguously identify your hits when you run a screen. This experiment is just shows the well-to-well variability. We take N of 12 or N of 16 and just run these controls and determine the Z prime. So in the enzyme reaction, easily you can produce a Z prime of 0.8, 0.7. I mean, a Z prime greater than 0.5 is considered really good for a biochemical screen. Now, moving to the TR Fred format, I'm sure you'd be wondering, is one format better than the other? And not really. They both perform quite similarly. And here is a titration of the same cGAS enzyme in the TR Fred format, and you get the exact same similar rates that you see with the FP assay. Once again, this is also nonlinear because it's a competitive binding assay where the cGAMP has to compete with the tracer and kick it off. That's what it means. And it's a nonlinear assay, and you can see the linearity when you run the standard curve. So both these assays perform very similarly. The choice would be completely dependent on the instrumentation that you have available in your lab, and I would say the ease, because some people are more familiar and comfortable with one format over the other. So that's the ease of comfortability from the users, and that's what will determine the assay that you choose. The Z prime, again, very similar Z primes that you get with both the assay formats. Okay, so we went ahead and determined the KM to make sure that we are running these assays under the standard conditions. And there are different ways of determining KM. In this particular example, we kept the GTP at 300 micromolar, DNA at 100 nanomolar, and determined the KM for ATP, which is about 100 micromolar, and that is well in the range of what's published in the literature, between 90 to 150. And again, it depends upon the kind of assays that has been used and kind of DNA that you also use, whether, we use ISD in this case, but it differs from, are you using a full length, 45 kilobase base pair DNA or just 45 base pair DNA. Moving to KM for GTP, again, you can determine that we have used these conditions, and we got a KM around 72 micromolar for GTP. And if you are interested in a protocol as to how to determine the KM using Transcreener assay, we have some application notes and resources in our website. So please check that out, and that goes through the steps how to do these experiments. Again, KM determination of DNA, as I said, you can try salmon sperm DNA versus, a smaller DNA, a 45 base pair interferon stimulating DNA, and you get the x-axis is in microgram per ml. So, the KM is either 0.9 or 0.04, depending on the kind of DNA. But the point being, you can do these kind of enzymology using the Transcreener cGAS assay. Substrate and DNA dependence. So this experiment was just to, before when we were commercializing, we wanted to make sure we are using the right kind of DNA to commercialize the product. And one is the interferon stimulating DNA. That's kind of what we have in the kit right now, but we also tested HSV. This is the herpes viral DNA. It is a 60 base pair DNA. So both these DNA perform well, but looking at the literature, most of the literature uses ISD 45 as the standard DNA that's used for cGAS assay. So we decided to go with that while commercializing this product. The graph here just shows that you need both the substrates, GTP and ATP, to activate the cGAS. Just having GTP or ATP alone by itself is not enough to get a response in the cGAS assay. So that's what this graph here shows. So with all that determined, so we determined the ideal concentration of cGAS that we were going to use. We confirmed that 100 micromolar ATP and GTP and 60 nanomolar DNA was a good standard condition to identify, both ATP competitive inhibitors or GTP competitive inhibitors, or even inhibitors that probably compete with DNA. So with those conditions, we thought we did a pilot screen here before doing our really huge screen for internal discovery program, but we just started with a pilot screen, and we used the Life Chemicals Diversity Library, which was just a very small subset of 1,600 compounds. The screen was performed at 10 micromolar, and we used cGAMP as at 0.8 micromolar as our control. It sort of produces around 10% conversion, the same amount of MP shift that we get. So this is all these black dots are the 1,600 compounds that were tested, and here are all the controls. So this is cGAMP at 25 micromolar. So this is like really saturating concentration of cGAMP, as you can see with the lower number. cGAMP at 0.8 micromolar. Here is what we would expect our compounds to be, and here is cGAMP at 10 nanomolar. These are the controls with no DNA here, and we did identify a couple hits. in this pilot screen. If you look at the table right below, the Z factor is about 0.62, and the Z prime is depending upon the concentration of cGAMP, it looks pretty good. Okay, so we have since then we have done for our internal program, we have done several big screens and identified some compounds, and here are some dose response curves that compares the FP and the TR-FRET assay. So as you can see, both these compounds give a decent hill slope close to 1, and it's showing negative because it's a negative kind of an assay. That's why the hill slope is has a negative number to it. But if you look at the IC50, we get like 1 micromolar with both FP and TR-FRET, and about 4.6, around 4.6 micromolar. So it's where both these assays perform very, very similarly. Okay, so shifting gears just a little bit. So as we do SAR here, we also test them with mouse cGAS just to see how selective these compounds are. Do they just inhibit the human cGAS, or do they also inhibit the mouse cGAS? So we will be, we are almost there. I think we're going to start selling the mouse cGAS very soon, but here are some preliminary data for it. So this is like titration of mouse cGAS. When we say complete reaction, we mean ATP, GTP, and the double-stranded DNA. We use the same conditions as we use for the human cGAS and see almost nothing, no cross reactivity in the absence of substrate. It's pretty clean. Again, it's nonlinear, and so you see, you run a standard curve and convert it. But what is different, as published in the literature, we see that the rate of the reaction with mouse cGAS is way higher than what we would expect for human cGAS, and that's been reported in the literature, and that's what we see here. Again, we do test some of our compounds. We test all our compounds against mouse cGAS, but this is just an example of a compound that showed about 28 nanomolar IC50 with the mouse cGAS. So, just showing that you could do all sorts of things with the mouse cGAS that we could do with the human cGAS. Okay, so at the beginning of when we were looking at the agenda slide, we did talk about applications. So yes, of course, you can run a screen, run dose response curves, but the more interesting ones are mechanistic studies to see, if your compound is an ATP competitive inhibitor or is it a GTP competitive inhibitor. So the way we set these experiments are just set the titration. So our standard condition is the one that's shown in yellow here with 100 micromolar ATP. But if you're trying to find out if your compound is competing with ATP, all you do is you vary your ATP. And you do not have to follow it exactly this way. There are so many different ways to do it. You can just run 100 and 1,000 or just 30 and 500. It doesn't matter. But we show here 4 different concentrations of ATP while maintaining the GTP at saturating concentrations, 500 micromolar GTP. We also keep the DNA at saturating amounts, about 500 nanomolar DNA. And you can see the IC50 here changes from about 1 micromolar to 7 micromolar, so about a 7-fold shift in ATP in the IC50. So you can plot just the IC50 in the y-axis with substrate over Km to see a linear dependence. And this compound perhaps would be, it could be an ATP competitive inhibitor because, it's not a very big, huge shift. I mean, we would like to see a shift more than 10-fold, but it's definitely trending in that direction. Now, the same compound, we want to test if it's a GTP competitive inhibitor. So again, you vary the GTP concentration here, 30, 100, 500, while keeping the ATP at high concentration. And here, the IC50 doesn't shift much. It shifts from about 1 to 3 micromolar, but if people who run these dose response curves would agree with me, like a 2-fold shift in IC50 is really common. It's a day-to-day variation. It's kind of the scatter in IC50 measurements. So a change between 1 to 3 is not considered very significant to kind of perhaps, to be a little bit significant, we need to at least see a greater than 5-fold shift between two concentrations. At least that's according to Robert Copeland's recent publication. So this we would say it's not GTP competitive, but this compound perhaps is ATP competitive. We have so far never identified any DNA competitive inhibitors in our SAR work here, but you could definitely do that kind of experiments too. All you would do is vary your DNA, from 30 nanomolar, 100 nanomolar, 500, and 1 micromolar DNA, and keep the ATP and GTP at 500 micromolar saturating, and you can see if the IC50 would shift with respect to DNA. So these kind of fun mechanistic studies are very easy to do and perform with the biochemical transcreener cGAS assay. And initially I had mentioned when we were talking about the assay principle itself, the antibody has a very, very quick equilibration time, meaning it can kick off the tracer and the cGAMP can bind to it in a matter of 5 to 10 minutes, which enables us to run assays in kinetic mode or in real-time mode. One of the applications of that came out of that is to determine the residence time or the K-off values. Now, the way to run these experiments, it's fairly simple. So you have already determined the IC50 of these compounds. Let's take this compound as an example, 352. Let's assume the IC50 is around 1 micromolar. All you would do is multiply that number, 1 micromolar, by 10. So let's put 10 micromolar compound, and the amount of enzyme that we typically use with our assay is about 20 nanomolar cGAS. Multiply that by 100, okay? So 20 times 100, 20 nanomolar times 100, 2 micromolar. So you take 2 micromolar cGAS and 10 micromolar of this compound, mix them together. So they are forming this EI complex. The enzyme is binding to the inhibitor. Give them some time, 30 minutes, 60 minutes. I do it for 60 minutes. 30 minutes should work as well. So keep the E and the I together, enzyme and the inhibitor complex together for 60 minutes. And then what we do is what is called jump dilution. So you do 100-fold dilution such that your 10x inhibitor now becomes 0.1 times the inhibitor, and your 100 times enzyme just becomes 1 times enzyme. So what it does is basically dilutes out the inhibitor from the enzyme, and then you start monitoring the recovery of the enzyme activity over time. So this black trace that you see is no compound. So you can see that when we have no compound, we just incubate it with DMSO, which is the solvent here. You can see the enzyme activity just recovers right away when there is nothing bound to it. But when these compounds are diluted, you start seeing a curvilinear graphs here. And although, the red trace shows that the compound does come off eventually, it's not bound for a very long time, which is like about 7 minutes or so. But if you look at the magenta trace here, which represents this compound 260, it's bound for a long time. So what it means is it's still engaging the target. So the recovery of the enzyme activity is kind of slow. The data analysis is a little tricky for residence time, but you can just use raw data and follow the time as they come off, and also, rank order your compounds based on how long they are bound to the target. Now that's and this is a very inexpensive way and a very, very quick way of determining the residence time of any of the compounds, as opposed to SPR or the other biophysical methods, which are more expensive and more time consuming. So you can do this in a high throughput fashion in a 384, or I do them actually, these kind of experiments, I do them in a 96 well-HAP area plate that helps me do 100-fold dilution by taking 0.5 microliters to a bigger volume. So I'm not pipetting very, very tiny amounts of EI complex. So we can run this in still it's a pretty high throughput format. We can do 96 compounds at one time. So that's another great application of this Transcreener cGAS assay. Now, we always conclude our talk or webinar with this slide that shows the assays that we sell. We have Transcreener ADP assay, Transcreener UDP assay, GDP assay, AMP, GMP assay. We call it AMP, GMP because the antibody binds to both of these nucleotides with the same affinity. We also have Epigen assays. We have assays for determining adenosine, and now we have cGAMP, determining cGAMP assays in both FP and TR-FRET. Now, even though it looks like we just sell a handful of these assays, but if you look at the kinases, the targets that we can just screen and study using these universal assays, you can study any kind of kinases and also other ATP utilizing enzymes like ATPases, helicases, heat shock proteins, carboxylases, so any ATP utilizing enzymes, and same with any of the targets that you take here. So you can study the breadth and depth of the of different targets by using a handful of these assays. And our website has like we have different assays that we sell, and please check out our website if you haven't already. There is a lot of data, a lot of resources and information, and of course we are always available should you be interested in any of these assays listed here. So here is, since you have all joined for the cGAS assay, I think this is what would probably interest you more. These are the catalog numbers or part numbers for the assays that we discussed today, the cGAS FP assay, cGAS TR-FRET assay. We also sell human cGAS enzyme, and very soon we are going to be selling mouse cGAS enzyme also. It's coming soon, so please check our website and stay tuned. It should be here. With that, I really thank you for listening to me and joining us today, and I'll take any questions that you have. Thank you.
All right. I have a few questions here, Mira. You showed some enzyme titrations with both FP and TR-FRET. When setting up a screen, what enzyme concentration would you suggest?
That's a great question. So the easy answer to that is if you buy the Bellbrook Labs enzyme, I would say anywhere between, we use it at 20 nanomolar, anywhere between 10 to 20 nanomolar cGAS should give a great response in both FP and TR-FRET. However, there may be customers who probably would use their own enzyme that they made in their lab. You will just need to validate that enzyme, and that is quite simple. You run the exact similar enzyme titration like I showed with ATP, GTP, double-stranded DNA, and no substrate control, and pick an enzyme concentration that would give at least Z prime around 0.5 or 100 MP shift. Just make sure that you're maintaining the linearity, which you should, because that's how we have designed our assays.
Okay, what is the range of cGAMP you can detect, and is there anything you need to do to tune the dynamic range?
That's a great question, because with some of our Transcreener assays, you do have to fine tune it because the antibody binds to for example, if we take a kinase reaction, there is ATP and you're producing ADP. The antibody that we generated is against ADP, but it still cross-reacts a little bit with ATP. So you do have to fine-tune it a little bit. Don't have to do that with this particular assay, because there is absolutely no cross-reactivity with ATP or GTP or DNA. So you could just look at the tech manual, and I think we say about 1 microgram per ml for the FP assay and about 4 nanomolar tracer, or 4 nanomolar tracer for the TR-FRET assay. And you can detect anywhere between 0.2 micromolar to, I mean, higher end is never an issue, but you can go as low as 0.2 micromolar, and you will still be in the initial velocity range. So that the assay itself has been designed to be under initial velocity conditions. So no fine tuning required for this particular assay.
Okay. Is it possible to detect cGAMP in lysates or blood and/or serum samples?
Not really. Well, let me rephrase that. Not yet. We have tried the assay in its current configuration, and it is not sensitive enough to detect in lysate. But stay tuned. We might be able to have something for you next year or so to detect cGAMP in lysate. So we're working on that.
If you want to use the assay for real time, will the conversion standard curve be needed at each time point?
No, because it is you're running a standard curve alongside. So, for example, you will run just one standard curve, and you will read the same plate, 5 minutes, 10 minutes. So you will have the standard curve also running in real time. But having said that, you don't really need to, because the antibody equilibrates so quickly that you could just have a standard curve at 60 minutes, like read at, and it should still work fine.
Okay, last question here. Is there any potential tips you have for running a transcreener assay like this? Any common questions that come up from time to time?
Yes, I do get the same questions all the time, and it is about the ceiling going down. So let me define ceiling here. So when you have, like, remember when I showed you let's take fluorescent polarization as an example. When you have cGAMP, you see, say 100 micromolar cGAMP, you saw a number that showed 24 polarization units, and when you had no cGAS, it was about 260. Now, in this reaction, you have ATP and GTP and everything in between. And let's say there is some nuclease in your buffer, okay, that is breaking down the nucleotides, and then the ceiling might slowly go down from 260 to 250 to 240. Doesn't happen very often, but sometimes it does. And most often, it is a reason because the buffer integrity uh, falls apart. So there is either a nuclease contamination, or some spores growing in your buffers. I mean, happens from time to time. So the best thing to do would be to make fresh buffers, use nuclease-free water where possible, and ATP and GTP, I mean, both of them kind of break down over time. I mean, they undergo non-specific hydrolysis. ATP not as much. GTP is prone. It's known for that. It kind of falls apart. We typically store our GTP in -80. At least store them in -20, and if possible, aliquot them and don't freeze thaw it more than 5 times. GTP is kind of a finicky thing. If you're using the terbium labeled antibody, the antibody that's in its native form, example, the FP assay, is kind of rugged. But when you have terbium, it's a little bit finicky because you have attached something to it. It doesn't, like to be mistreated. So I would again say, good lab practices are always helpful. Aliquoting, not, so you don't have to freeze thaw it more than 4 or 5 times. And even if you aliquot, don't aliquot 1 microliter or 2 because of the difference in the air liquid interface. So, at least have 5 to 10 microliters as the minimal volume when you aliquot and freeze it. If you do have to freeze thaw it, I would recommend not. The FP, we generally say 5 times, but the terbium, I'd be really careful about that. Yeah, that's what I would have to add.
Great. Thank you, Mira. That's all the time we have for today. With any other questions, please contact us via email at info@bellbrooklabs.com. Thank you for joining us. A copy of the webinar will be sent within the next 24 hours.
Thank you.